Welcome back to this course on organic chemistry in biology and drug development. We have already completed the first part of this course, that is organic chemistry and biology, and we have read that amount of biology which is required for understanding the drug discovery and development process. Okay. And we have started the drug discovery or development processes by pointing out that what is the practice nowadays to ultimately come up with a drug, and there are several sequence of steps, that is identification of the target, validation of the target, hit identification, lead identification, then lead optimization, followed by PK/PD studies, that is the pharmacokinetic and pharmacodynamic studies, then the toxicological studies, and the preclinical, that also includes the preclinical studies, that is also a part of the preclinical studies, which is basically studies on animals or sometimes in other in vivo studies. It could be with cells. You can determine the toxicity, cytotoxicity, etc. And once that is through, then the drug goes for the human trials, that is the clinical trials, and there are different phases also, phase 1, phase 2, phase 3, and once it is approved, then subsequent years the pharmaceutical companies also look at the activity or any side effect that the drug can have as a long term side effect. So, after it is introduction to the market, so the subsequent years they also follow the effect, and that is called phase 4 clinical trial. Okay. So, that is there. Then we started and we completed the combinatorial chemistry, that means how to arrive at a quick hit compound by the process which is basically generation a large library of compounds, and then via high throughput screening you can pin down a hit compound quite rapidly. Okay. That is what is the domain of combinatorial chemistry. And then we started the actual medicinal chemistry, that means the target oriented discovery of compounds which are acting as hits. So, the first one we talked about is the neurotransmitters, and we have covered acetylcholine as the first neurotransmitter. We have seen that the acetylcholine can have two receptors, muscarinic receptors and as well as nicotinic receptors, and they are entirely different. One is ligand gated ion channel and the other is the G protein coupled receptors. And there is an enzyme called acetylcholinesterase, which is very important because that keeps the concentration of free acetylcholine versus the bound acetylcholine. Bound means bound with the receptor, that optimum concentration is maintained. So, if there is a problem with your acetylcholine esterase, if it is blocked, then you have a problem. So, there are apart from the drugs, there are antidoses to generate acetylcholine esterase, especially when there is poisoning by the nerve gas type compounds, or if somebody has consumed any organophosphorus which are present, which are acting as an insecticide compounds. Okay. So, that part we have covered. Now, today we will discuss another neurotransmitter or another two neurotransmitters. One is called the aromatic, the catechol based neurotransmitters. Okay. Catechol based neurotransmitters. That means they have a catechol moiety. What is the catechol moiety? That is orthodihydroxy aromatic ring, catechol based neurotransmitters. Okay. Now, the first class is what is called in this class these compounds belong to, one is called this dopamine, that is you see dihydroxy phenyl ethyl amine, and also you have noradrenaline, and the third one is adrenaline. By the way, adrenaline is a hormone, but these are neurotransmitters. You know the difference between the hormone and the neurotransmitter, that basically these are acting at a very short distance. Noradrenaline and dopamine also belongs to the class of a neurotransmitter. Okay. So, whenever we study neurotransmitters like this, and if there is a problem in the concentration of these neurotransmitters, then we have diseases. Now, to stop or to keep the concentration of the dopamine or noradrenaline or even adrenaline at the optimum level, so you have to know the biochemistry or the biosynthesis of these molecules. Once you know the biosynthesis, then there is a question of intervening in the process of production or destruction of these neurotransmitters. Okay. Because the disease is basically the destruction and the formation, there is a disbalance. Okay. Then why then you have the problem. Now, the question is what these neurotransmitters do in the body. So, the neurotransmitter dopamine is written here, is a primary endogenous, that means inside, endogenous ligand for dopamine receptors. Like all neurotransmitters work by binding to a receptor, but these receptors are very characteristic of the type of neurotransmitter that you have. Like acetylcholine, we know that cholinergic receptors, and then you have for dopamine, that will be called dopaminergic receptors. Okay. So, dopamine is the primary endogenous ligand for dopamine receptors. Dopamine receptors are implicated in many neurological processes. That means, when dopamine binds to these receptors, lot of signal transduction processes take place, and that ultimately results in motivation, pleasure, cognition, memory, learning, fine motor control, that means the way we move our hands and limbs, as well as the modulation of endocrine signaling, neuroendocrine signaling. Okay. That is basically the modulation of signaling, that is by the hormones generated from the endocrine glands. Okay, because the hormones are usually secreted from the glands. So, that is, so you see that there are so many effects that neurotransmitters like dopamine have. Okay. And then the other one is what is called norepinephrine, or this is earlier days it was known as noradrenaline. So, norepinephrine increases the heart rate, blood pressure, triggers the release of glucose from the energy stores, increases blood flow to skeletal muscle, reduces blood flow to the gastrointestinal system, and inhibits voiding the bladder and gastrointestinal motility. So, here the very important part is that it increases the heart rate and blood pressure. Okay. So, it must be the causative agent. If somebody is suffering from hypertension, of course, hypertension has different mechanism by which the blood pressure can rise, but one of the mechanism is what is norepinephrine related. Okay. Now, let us see how this dopamine is generated, dopamine and norepinephrine is generated inside the in the neurons. Okay. Now, what happens, the starting point is a simple amino acid which is called L-tyrosine. Okay. Now, tyrosine is, you know, is a protein building amino acids. So, apart from the amino acid function, this has another role, very important role in generating the dopamine and norepinephrine and finally adrenaline, or that is called epinephrine only. Okay, here it is adrenaline. So, that is also called this is noradrenaline, this is also called norepinephrine, epinephrine, and this is called epinephrine. The same thing, but the more accepted or is the adrenaline. We immediately understand adrenaline and noradrenaline. Okay. So, when we get excited, like during an exam, we have the more of the adrenaline secretion there, so that you sustain to the stressed condition. So, whenever there is any stress, then you have this more secretion of adrenaline, and basically that means you have more secretion of noradrenaline. Now, let us see, L-tyrosine is the starting point. Now, L-tyrosine is an amino acid which is shown here, the structure. Now, what happens that the brain cells, that means the neurons, get this metabolite like L-tyrosine. If I take L-tyrosine orally, then the L-tyrosine will be absorbed from the gastrointestinal tract, and then it will be circulated in the blood. Okay, it goes into the blood and that circulates. Now, the question is, when something is in the blood, that does not mean that it is, although the blood goes to the brain, but the drug may not be transferred to the cell, to the neuronal cell directly. There is what is called a barrier from blood to brain, and that is called the blood brain barrier. Not all molecules reach the brain from the blood. Okay. And L-tyrosine reaches the or crosses the blood brain barrier and then reaches the brain. Now, the process by which this crossing takes place is usually a carrier molecule is there which takes L-tyrosine and transfers it to the crosses the blood brain barrier and puts the tyrosine here inside the brain cells. Okay. So, this is your tyrosine. So, as it reaches the brain, now there is an enzyme which is called tyrosine hydroxylase. Tyrosine hydroxylase, which actually which puts a hydroxy group in the aromatic ring, and that is basically the catechol moiety. Now, you have the catechol moiety, and so basically now you have hydroxy dihydroxy. See, tyrosine you can call as hydroxyphenylalanine, and this will be dihydroxyphenylalanine. Okay. So, that is what is abbreviated as L-DOPA, that means dihydroxyphenylalanine, L-DOPA. Okay. Now, L-DOPA then undergoes a decarboxylation. So, obviously, this will be a PLP mediated decarboxylation, because we know alpha amino acids undergo decarboxylation via enzyme which is dependent on the pyridoxal phosphate. So, the CO2, after it loses CO2, it generates what is called dopamine. Okay. And then dopamine undergoes another hydroxylation. This is the beta hydroxylation, because this is your alpha carbon from the starting amino acid, this is your beta carbon. So, you get beta hydroxylation. So, once you get beta hydroxylation, that gives you what is called noradrenaline or norepinephrine, and then that undergoes a methylation, the amine gets methylation, and so it becomes NHME, that is what is called adrenaline. Okay. So, that is adrenaline, this is noradrenaline, then you have dopamine, it is backwards L-DOPA, but the starting point is L-tyrosine crossing the blood brain barrier. Okay. Now, this L-tyrosine, now in some diseases where problem is there will be less production of this dopamine in many of these say depression, because dopamine gives you pleasure, cognitive behavior, then motor your motor functions, etc. So, maintaining a concentration of dopamine is very important. Now, how the concentration can be low? There are pathways. See, first of all, either it may not be produced in the, so the enzymes may not be very active. So, it is not produced in large amount. The other is, there are enzymes which are called MAO enzymes, MAO, monoamine oxidase. What it does? It destroys the primary amines if they are generated. This is called monoamine oxidase, monoamine oxidase. So, the concentration of dopamine, monoamine oxidase, sorry. So, there are two ways by which dopamine concentration can be low. One is that the biosynthetic machinery is not functioning properly, that is number one, and number two is that the monoamine oxidase may be overactive, and that is causing the destruction of the primary amine, which is there in dopamine, which is there in norepinephrine. So, these are the two processes. Okay. Now, monoamine oxidase, you just know it, we are not discussing monoamine oxidase. What we are discussing that if we see that there is a problem in the biosynthetic machinery, then how to increase the concentration of this catechol amines. Okay, this is a catechol amine. One way is that you take more of tyrosine. Suppose the tyrosine is not present in large amount. So, if you take more of tyrosine, then tyrosine crosses the blood-brain barrier and you maintain a higher concentration of tyrosine here. So, that will be converted into the dopamine. That is one way, but the problem is, if somebody takes tyrosine from outside, then tyrosine being a protein amino acid, so its main function that it will be used to make the proteins that are required in the body, because tyrosine is a protein amino acid. So, by the time everything reaches to the brain, that will be very less, because most of the tyrosine will be utilized to make the proteins. So, that is not a viable treatment, that if you take too much tyrosine. So, that is gone. The next is, you can have a very direct approach, that dopamine is a very simple compound, you can make it in large quantities, but the problem is, if you take dopamine orally, the dopamine cannot cross the blood-brain barrier. I will show you here. So, this is what is the basically what we are talking about. Periphery is that boundary. Periphery basically is just outside the boundary of the blood vessel. So, L-DOPA, you see here it is written that tyrosine crosses, I told you, but tyrosine is not the answer to treat this type of diseases, because tyrosine will be utilized mostly as the primary role, that is to make the proteins. L-DOPA definitely is not used, it is not a protein amino acids. So, you can take large doses of L-DOPA, and that also can cross the blood-brain barrier. So, that has the ability to cross the blood-brain barrier. So, this is a viable alternative for treatment of low dopamine concentration, that you take L-DOPA. So, that should cross the blood-brain barrier, and then the enzyme, that is the decarboxylase enzyme, which should DOPA decarboxylase, that should convert it to the dopamine, and then dopamine produces the effect that is required. But the problem is, L-DOPA is also an amino acid, and again here, by the time it reaches reaches near the blood-brain barrier, there is this decarboxylase enzymes which decarboxylate DOPA into dopamine out before it has reached the brain cells. That means, what I am saying, that if we suppose this is a blood vessel and the neuronal cells are somewhere near here, and this is the peripheral region, okay. And what happens, that L-DOPA goes in the bloodstream, and L-DOPA is mostly decarboxylated into dopamine before it reaches the brain cells. So, if it is already converted to dopamine, problem is dopamine cannot cross the blood brain barrier. So, dopamine will not enter the brain cells if you use the L-DOPA, that is a problem. Of course, you can say that some of it definitely goes into the brain, but that may not be very optimum concentration. Okay. Now, to bypass this route, so now we have learned three things. L-tyrosine is not the answer, L-DOPA is a partial answer, that some portion of it goes into the brain, crosses the blood brain barrier and then formation of dopamine, but a majority of it is decarboxylated into dopamine before it if it is exposed to the barrier. So, dopamine cannot cross it. So, most of it is remaining outside the brain cells, okay. And this dopamine undergoes degradation by, as I said, that it will be, there are many monoamine oxidases that will dissociate, that will degrade the dopamine. So, to bypass this, what you need to do is to utilize inhibitor of this DOPA decarboxylase. This is peripheral DOPA decarboxylase, and this is not the peripheral, this is in the brain cells. So, on this side is brain, on this side is the periphery. So, it is the peripheral DOPA decarboxylase that is the problem. So, what you do, you along with L-DOPA, what you can do, you add inhibitors of this DOPA decarboxylase, and this does not cross blood brain barrier. So, if there is any, it can only inhibit the peripheral DOPA, L-DOPA decarboxylation. So, you stop this decarboxylation, then most of the L-DOPA will be on this side. Okay. The equilibrium is shifted now. So, that will create a high concentration of L-DOPA, and then that will be converted into dopamine, and you get the desired effect. Okay. So, these are some of the compounds which act as your 64 means this compound benserazide, that is a DOPA decarboxylase inhibitor, and then you have a very similar kind of inhibitor which is called methyl DOPA. The structure is not given here. Methyl means there is alpha methyl. So, at the alpha carbon you have a methyl group. I think I have the structure somewhere, carbidopa. So, not methyl DOPA. Methyl DOPA is also another compound which you can give to regulate the blood pressure, because methyl DOPA is not the it has got a methyl group at the alpha position, and that creates an antagonist type of effect. So, that the blood pressure which is associated with norepinephrine and adrenaline or epinephrine. So, that blood pressure can be controlled. In fact, the earlier days, one of the good medicine was this M-DOPA for controlling the blood pressure. The other important thing I forgot to tell you is that I told you about these motor functions that it controls, dopamine. Now, dopamine also controls I can show you the dopamine also controls the motor function, motor control, and this is what is there in the case of Parkinson's disease. Parkinson's disease is a very bad disease, Parkinson's disease, where people lose the motor functions. They cannot even move their hands or limbs. Also, they cannot remember things, because there is this cognition behavior that also goes off. Memory also is not there. So, that is what is called Parkinson's disease, and the only way the Parkinson's disease can be treated is to utilize this L-DOPA is the only compound, L-DOPA along with the peripheral DOPA decarboxylase. Remember, then you can use a lesser dose of L-DOPA, and that creates the concentration of dopamine to some optimum level. Okay. However, Parkinson's disease is you can slow down the Parkinson's disease, but it is not possible to cure the Parkinson's disease, because whatever genetic machinery is defective, that you cannot repair. What you are doing, just trying from the external source, trying to maintain the concentration of dopamine. Okay. So, it is not a cure, but it is a way to slow down the onset, slow down the progress of the Parkinson disease. So, this is a very important, dopamine is very important, or these catecholamines are very important neurotransmitters. These are associated with Parkinson's disease, and also the mood fluctuations, because it gives pleasure, it controls your mood. So, that is also very important. So, that is what is all about the dopamine chemistry. Now, let us talk about one more, which we have already told you earlier. It came into the into our discussion when we said that many of the amino acids that are present in the body, they may not be alpha amino acids, and one of the they are not part of the protein that we make, but they have a distinct function mainly as neurotransmitters. Like glutamic acid also, like tyrosine is a precursor of neurotransmitter. Glutamic acid itself is a neurotransmitter. Okay. And then from glutamic acid, again there are amino acids which are generated. One of them is what is called the GABA, or the gamma aminobutyric acid. Remember that we have discussed that when we were talking about the PLP, the chemistry of PLP. So, we have gamma aminobutyric acid. So, this is the compound, very simple compound, and its biosynthesis is obviously from the glutamic acid. So, if you have L-glutamic acid, so CH2, then CH, NH2, CO2H, and then CH2, CH 1, 2, 3, 4, 5. So, it is CO2H. So, that undergoes decarboxylation. So, this is called glutamate decarboxylase, because it is decarboxylating a glutamic acid. So, what you get is what is called GABA. Okay. Now, GABA, I told you at that time, this is called an inhibitory neurotransmitter. See, we have both types of neurotransmitter. One is excitatory, another is inhibitory, and we have to have both, because all the time we cannot be excited all the time, because that also is not good, because your blood pressure goes up, norepinephrine will be more. So, blood pressure goes up. So, there must be something which also is inhibitory. So, it also plays down or controls the effect of neuroexcitation. Okay. So, this is an inhibitory neurotransmitter. Now, low levels is implicated in diseases like what is this convulsion, that is called Briggy, that means in Bengali that has got a name. The other is that convulsion, it causes convulsion, that means the person loses its sense, and that is what is called convulsion. I think there is a name for that. I will, if I remember that, I will tell you, but some people have this problem that occasionally they lose their sense and become they lose they are not basically they lose everything. It appears that the people is almost dying. So, loses the consciousness, and then that is what is called convulsion. Yes, now I know it is epilepsy. Okay. Epilepsy is the disease which causes convulsion at certain point of time, and nobody knows why, at what time it will strike a person. Okay. Now, epilepsy is related to the lowering of the concentration of GABA, the gamma aminobutyric acid. Okay. Now, I already told you the biosynthesis of GABA, that means it comes from here glutamic acid. So, glutamic acid causes the blood brain barrier, and then it undergoes the glutamate decarboxylase and GABA is formed. Now, how the concentration of GABA is maintained. See, this is very important. You need to know not only how it is generated, you also need to know how it is degraded. Like acetylcholine is generated, and then its degradation was by the enzyme acetylcholine esterase. Dopamine is generated from L-tyrosine, its degradation is from by monoamine oxidase. Then you have this GABA, its formation is controlled by the glutamate decarboxylase, but the question is how it is degraded. Okay. So, before we go on to the any medicinal aspect of this, because obviously, if you can increase the concentration of GABA, that is going to act as an anti-epileptic agent. Okay. How GABA is question is how GABA is degraded. Okay. So, again I write, you have glutamic acid like this, and that undergoes glutamate decarboxylase, and you form the gamma aminobutyric acid. So, this is what is GABA, and then this GABA undergoes transamination. GABA that is called GABA T. GABA T means gamma aminobutyric acid transaminase. That means what is transamination? That is also a pyridoxal, you have read already, pyridoxal mediated reaction, where amine from one compound is transferred to an alpha keto acid, removing the carbonyl and replacing that with amine, and in the process the original compound, which is an amine donor, that is converted into a carbonyl compound. Okay. So, here this GABA, the corresponding alpha keto acid is the pyruvic acid. So, any transaminase will have two components, one is an alpha keto acid, another is the amino acid. Okay. It is not necessarily it will be alpha amino acid, here it is a gamma aminobutyric acid. Okay. So, what will be the reaction? The keto acid will be converted into what? NH2 CO2H. So, this is nothing but alanine, and what will happen to the GABA? That will be CO2H, and then that will, instead of NH2 you will have a carbonyl. So, this is the compound, that means NH2, remove the NH2, put a carbonyl, but remember there is a hydrogen here also. So, that is called succinic semialdehyde. Semialdehyde means succinic acid has two carboxylic semialdehyde. So, you can write, actually you can remove this word, succinic semialdehyde. So, one of the carboxy is converted into aldehyde. So, this is how the GABA is degraded. Now, if there is a low concentration of GABA, so now you have two choices, either you add more glutamic acid, so that you get more concentration of GABA, or you can stop this GABA transaminase by using an inhibitor, so that GABA is not degraded rapidly into alanine and succinic semialdehyde. Okay. So, the first one will not work like L-tyrosine. If you give L-tyrosine, you are not going to get any high concentration of dopamine, because tyrosine is a part of the protein amino acid. So, its major function will be to utilize in the formation of the protein. Similarly, glutamic acid is like that. Glutamic acid is a part of your protein amino acid. So, that will also be consumed, mostly consumed in making the protein amino acid. So, that part will not work. So, what will work is can work is this GABA transaminase, that if you can form an inhibitor. So, the treatment is basically finding an inhibitor of GABA transaminase. Okay. So, now you see your earlier knowledge about biochemistry is directly now relevant, becoming relevant in understanding the medicinal chemistry, the genesis of different drugs and the strategies that you have to adopt to make those. Now, let us talk about this GABA transaminase inhibitors. Okay. So, one compound is what is called gamma vinyl GABA, gamma vinyl GABA. So, this is your GABA. So, very similar compound, but you put a double bond here, a vinyl. So, this is called gamma vinyl GABA. Now, this is an inhibitor and the mechanism will be, remember the transamination reaction involves reaction with pyridoxal phosphate. This pyridoxal phosphate, you should remember the structure. This is a CH2OP and this is a OH and that is a methyl. So, methyl, this is OH and this is OP, that is called pyridoxal phosphate, and in the biological system it is present as in the protonated form. Okay. So, now what will happen? Free amines will definitely react and form the imine. So, that forms the imine. Imine with pyridoxal, sorry, not this one. So, that will form the imine. So, CH and then the pyridine nucleus NH plus and then double bond, and we are not writing the substituents, we are writing this because it is tri-substituted system. Okay. So, once that is formed, you know that this is an electron sink because of the positive charge. So, what will happen? This hydrogen, alpha hydrogen, because you know that the chemistry is dictated by either loss of hydrogen, loss of carbon dioxide. These are the two reactions. So, here there is no CO2H. So, the hydrogen will be lost and this goes here, goes up to the nitrogen. So, you have CO2H and then you have this double bond, you have a double bond here N and you have CH, and then the pyridine is no longer aromatic. So, it will be like this. Now, it becomes, it regains the aromaticity. It regains the aromaticity by pushing back again, but the hydrogen that is taken up is at the CH2. So, what you will get is a CO2H in CH2 and then the pyridinium system NH plus here, 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 and this is a double bond. Now, let us see, this is an iminium ion. So, this is nothing but this is resembling an alpha beta unsaturated carbonyl system where the oxygen is replaced by nitrogen. And what is the usual reaction of this alpha beta unsaturated systems attached to an electron withdrawing group is what is called Michael reaction, that if you have a nucleophile, now the nucleophile can attack here and can add to it by what is called Michael addition. So, now this molecule is well placed to accept, to have to act as an acceptor of a nucleophile via Michael addition. So, remember these reactions are actually going on in the active site of an enzyme. So, if the enzyme has an electron electron rich group XH, it could be SH, it could be OH. So, then the, or it could be an NH also, NH2 in the form of not, which is not present as NH3 plus, then this XH can add here. So, what the XH can do, it can add to this double bond and forms a covalent connection. So, basically CO2H and then X will add here, and this is your, that will be double bond here, and this N H and then CH2, and you have this pyridinium thing, okay, NH plus. Now, this will remain like that. Okay. So, basically it is a case of irreversible inhibition, because you have a covalent bond connecting to the cofactor. It is not the cofactor, it is the substrate. Okay. The substrate is hooked to the cofactor. So, everything is now blocked. So, if that be the case, that means this is a case of irreversible inhibition, and the question is whether it is an active site directed or is it a suicide inhibition. You see, this is a suicide inhibition because the normal reactions proceeded up to certain extent, and that creates a reactive system. So, this is nothing but what is called suicide inhibition. I remember again, I just remind you that suicide inhibition is the one where the substrate is processed to some extent, and then it generates a very reactive compound, and to which the new enzyme side chain reacts. So, that is what is the case here. So, it is an irreversible inhibition, not only irreversible inhibition, it is a suicide inhibition. So, this is a basic it is approved as a drug to treat the epilepsy, because you are inhibiting that transamination enzyme, okay, the transaminase enzyme. Similarly, you have another compound, let us see, I think that will be here. Similarly, you have another compound which is called which is this compound. It is an aromatic, it is not an aromatic, it is a dihydroaromatic compound, because that is saturated. Okay. So, it will have a great tendency if there is a scope of forming a double bond there. So, that is so let us see how it works. So, you have this compound, you see this is almost like a GABA amino acid, but it is hooked up to this diene. Now, what will happen? I can show it here. First, this pyridoxal phosphate reacts, forming the imine, and very similar that it acts as an electron sink, and the electron flows here up to this point. So, you have the imine here, and then as soon as the imine here, like last time, like vinyl GABA, the adjacent there is this now in GABA, in gamma phenyl GABA, you get a alpha beta unsaturated imine. So, there will be addition of the active site amino acid via Michael addition. Here what happens, this hydrogen will now be lost, and this double bond will come here. So, that means this is NH now, and there is a double bond here. Okay. So, now it has become an aromatic compound. So, if it has become an aromatic compound, now there is no way this pyridoxal phosphate can be freed from the molecule. So, this PLP gets covalently linked to this compound. This has got a name. I can just try to find out what is the name of this compound. Means as a, it is a drug, but I may have, I may not have. It has got a commercial name when it acts as a drug. Let us see whether the name is not here. No, no name is given. But name is not the most important issue. The most important issue is the mechanism. Okay. And I hope you will, I can tell you the name. I think it is there. This is gamma phenyl GABA, we have said, and the other compound what is called normal substrate gamma phenyl GABA, it is gabaculin, right. This is the commercial name is gabaculin. This is also, these two compounds are used as drugs to treat the epilepsy. And if somebody asks what is the mechanism, you can say that the mechanism is basically inhibiting by irreversibly inhibiting the transaminase enzyme. Now, just there is a slight difference in the mechanism of action of the two. GABA vinyl GABA is covalently linked to the enzyme as well as to the cofactor. Here it is linked to the cofactor, but if you can neutralize the cofactor, that means the enzyme cannot act also, because the enzyme needs this cofactor to function as a transaminase enzyme. So, that is all about the three types of neurotransmitters we have covered. We have covered the acetylcholine, we have covered the dopamine, and we have covered the this GABA and its associated diseases and how to treat that diseases. Okay. Thank you.